Thanks for this opportunity to talk to you today about populations. Populations, like many words, is semantically overloaded, which is to say it's often used to mean many different things. For example, what I, as an ecologist, think of as a population, which is a collection of organisms of the same species living in the same place, is often referred to as by other researchers as a community. For example, people studying collections of humans, whether they're epidemiologists or sociologists, often refer to collections of humans as communities. Likewise, what I think of as a community, which is a multi-species collection of organisms interacting with one another and living in the same place, is often called a population by some people. Furthermore, with new sequencing methods, such as metagenomics, populations can now be defined using clustering algorithms and statistical methods. So with all these different ways of talking about populations, how can we create ontology classes that satisfy these many different needs? The PCO has taken a neutral approach to defining populations by specifying a top-level term called collections of organisms. A collection of organisms is defined as an object aggregate from the basic formal ontology that has as member two organisms or viruses or viroids. Throughout this talk, I'm going to use the term organism for brevity, but bear in mind that I include viruses and viroids within that term. So once we have this upper-level collection of organisms terms, we could then proceed to define different types of collections of organisms for many different needs and to satisfy many different researchers. Collections of organisms can be defined based on different characteristics, such as their composition. Are they made up of a single species or multiple species? Their interactions, are the members interacting with each other and how? Their location, for example, populations and ecological communities are expected to occur within the same location. Or even by their relatedness, such as in the collection called a family. Any number of other characters can also be used. PCO also includes terms for processes such as population extinction, population growth and migration, as well as terms for qualities of organisms such as carrying capacity, population birth and death rates, species richness, and species diversity. Note, I put the qualities in quotes here because these are not all strictly qualities in an ontological sense, but they are characteristics of the different collections of organisms being described. Another common use for the term population is in statistics. A population is, of course, the entire collection of organisms about which we want to know something, but we can only usually measure a subsample of them. And so some sampling process takes place, and then statistics are calculated on the sample. PCO is not a statistical ontology. It can be used to describe the population in a statistical population, provided that, of course, that population is made up of organisms. But it cannot be used to describe the sampling methods, which are better handled through other ontologies, such as the statistical methods ontology, or STATO. So for the remainder of the talk, I'm going to describe how this notion of collection of organisms and the PCO can be used in omics studies, giving two specific examples from environmental sampling and association studies. In each of these examples, I'll describe how using ontology classes can help to clarify the data and the terminology used in those studies. GWAS studies are a popular way, a common way of used currently to look for associations between genes and traits of interest. In a GWAS study, the genome-wide set of variants is examined in many different individuals to see if there are correlations between the trait and the gene sequence or the gene of interest. An important part of GWAS studies is controlling for population structure, that is, for the presence of subgroups within the population that have differences in ancestry. Failing to account for population structure can lead to false positives or spurious associations in GWAS studies because ancestry can actually lead to relationships between gene sequences, between DNA sequences and traits that are not actually due to controlling factors of the gene. So how will we describe population structure within the PCO? We start with our basic definition of a population, which is a collection of organisms, all of the same species, that live in the same place. And then we can create a subclass of that called structured population of organisms that has organismal subpopulations as part. And just as we describe different types of collections of organisms based on different criteria, we can also describe different types of subpopulations. In this case, however, the criteria will be things like clustering algorithms and statistical methods. They may, of course, however, be other factors such as geographic location or pedigree. Another use of populations, another important use of populations within omics is through environmental sampling and metagenomics. Environmental samples are specimens of materials collected directly from the environment, such as this ocean sediment sample being collected, shown here. Specimens are collected from different environmental materials are measured for their biological and environmental characteristics, but because it's actually difficult or even impossible to directly observe the organisms living within these specimens, even using microbial culturing methods, metagenomics, or the study of the genetic material recovered from these samples, has become the common way of studying their biological components. The goal of environmental studies is to identify relationships between the environment and the microbial community composition and function. So once the specimen has been collected, its environment is described using ontology terms such as the environmental material, feature, or biome from ENVO, but also using more quantitative terms or measurements like the temperature or dissolved organic carbon. In order to understand the biological community within the specimen, sequencing is carried out. As I mentioned, metagenomic sequencing is carried out. And then those sequences can be clustered into collections of OTUs, or operational taxonomic units. Using the PCO, using terms from PCO like ecological community and species diversity, these OTUs can then be used to describe the biotic aspects of the community. Other types of sequencing that are more comprehensive for the whole genome can also then be used from the same specimen to describe the molecular function or biological processes occurring within that community using terms from the gene ontology, or GO. One of the challenges in these studies is actually describing what's meant by these OTUs that are defined in metagenomic studies. The original definition of OTU from Sokol and Sneath back in the 70s was the group of organisms being studied. Very general. Within metagenomics, a slightly more specific version of that has been adopted. It's usually the collection of organisms grouped by their DNA sequence similarity of a specific marker gene, as I described in the previous slide. The key here is that both of these definitions are operational, so that means they can essentially be whatever anyone wants them to be. And different methods, of course, different clustering methods will create different OTUs. Different operations lead to different collections of organisms. And this creates challenges when you're trying to reuse or make comparisons across different studies. If the OTU definitions are not the same, then it makes it very difficult to compare two different studies. In order to enhance comparisons and make it possible to really fully understand the methods used in different studies, it's important to very explicitly define what happened as part of a study. So a few years ago, we did an exercise in which we fully mapped out an environmental sampling process using terms from different ontologies, including the Ontology for Biomedical Investigations, or OBI, PCO, PATO, a number of different ontologies here. And I've intentionally left this figure small and blurry so that you don't attempt to read it in this presentation. But if you're interested, please see the paper by Roca Serra et al. In Database from 2015. Here I just pulled out a few of the key classes to make the points for today's talk that describe this study. In this case, there was some soil, which is the environmental material being sampled, and then this the black dotted line represents a number of different processes, material handling processes that happen to that soil. The final one being a targeted gene survey, which is a very, very specific type of assay in OBI. and the output of that targeted gene survey is an OTU matrix. That OTU matrix can be used to calculate the diversity of some ecological community that was located in that soil specimen. Here, notice that used to calculate it is also shown as a dotted line, because at the time when this figure was made, the Stato ontology had not been released. Currently, we could now use very specific methods and ontology terms to describe exactly how diversity is calculated and also the type of OTU matrix. So what we would need to do is make specific subclasses or instances of targeted gene survey that correspond to different clustering methods used in metagenomics. So defining OTUs, it turns out, in fact, is actually fairly consistent with the PCO. Our top-level term of collection of organisms is essentially what an OTU is, the collection of organisms being studied, however it is you choose to define them. In the case of a microbial community, it's all of the microbes living within a particular area in a particular specimen. If you then use clustering methods to define OTUs that that are known to represent clades, that is, that those OTUs, the method of OTU generation provides groups that are based on evolutionary history, then you could use the PCO term population to describe that OTU. If you're using OTU clustering methods that do not necessarily, or if you do not necessarily lead to groups of organisms that are related evolutionarily, but simply have sequencing similarity, then you could say that you could still use PCO. It's just a question of defining your collection of organisms based on taxonomy rather than phylogeny. The real problem comes in is that many people are now using the word OTU to define clusters of sequences that may or may not actually have any relationship to organisms or population. In that case, PCO is not appropriate for describing those quote-unquote OTUs, but rather they should be described using terms from Stato as the outcome of some statistical process. So in conclusion, the PCO can be used to define classes for any collection of organisms, but it is important that the algorithms yield clusters that actually map to organisms in order to have meaningful OTUs. As the examples in this talk have shown, multiple ontologies are required to fully describe omics data, and this is one of the big advantages of using ontologies from the OBO Foundry, is that the different ontologies are readily interoperable with one another. I've already mentioned most of the ontologies that you'd most likely need to use when describing omics data: PCO, ENVO, the Gene Ontology, Ontology for Biomedical Investigations, BioCollections Ontology, and Stato. And I'll just take a moment to put in a plug for the new ontology EcoCore, which describes key roles and interactions in ecology. So with that, since I'm not there, I can't actually take any questions, but I'm sure that Pierre would be willing to answer questions as best he is able, and thanks so much for your time.